and terrestrial bacteria, fungi, and plants by applying liquid chromatography HRMS de-replication strategies. He has optimized downstream process for large-scale isolation of natural products and developed a methodology for fingerprinting of extracts with LC-UV-MS in natural products-based drug discovery program. Dr. Abhijit's research group is investigating lichens grown on mangroves from Gulf of Kutch and Gulf of Camber region of Gujarat to find bioactive molecules. Dr. Abhijit has 3 patents to his credit and more than 28 research publications from international journals. So with this short introduction, I invite Professor Abhijit to give us a talk. The floor is yours, Doctor.
Thank you, Dr. Murugan. And can you hear me all?
Yes, sir.
Great. Good morning, everyone. At the onset, I would like to thank Srishti and BIIS program for inviting me for this talk. And I would also like to thank Professor Anil Gupta for this invitation and all the team members of Srishti. Yes, so good morning. Today, I would prefer also to have an interactive session so that if you have any questions, you can ask me in between also, or at the end also, whichever way you are comfortable with. I'm fine with both. I will share my slides. Let me know if you can see those slides properly. Today, I will go through some of the understanding of natural product research and its contribution towards the medicine, and also like how analytical advancement have played an important role in this aspects. That will be the key area of my research also, and I will show some highlights of how that analytical tools have improved the research and output. So we will discuss that. So I'm sharing my slides. So let me know if you can see the slide properly.
Yes, Doctor, it is visible.
Yes, great. So let's get going. So as we discussed that we will talk about the analytical techniques in natural product research and what we refer them as de-replication, and we will see now how and why they are important. So just as a background so that we will make a context properly, natural products and nature, to be specific, have helped human being from so many years. I think there is no second thought about it, right? This is one of the milestone flow chart which I borrowed from one of the published literature here, that they have clearly highlighted a clear-cut milestone, the achievements of the natural product drug discovery program. I won't go back long, but you know, there were several Nobel Prizes that people have received, and the recent one was in 2015, where Professor Yu Tu has received for the discovery of artemisinin and ivermectins, right? And then recently in 2019, and say around that, the China has approved a seaweed-based oligomanate compound for the Alzheimer drug, which is also suggesting that the people are still working on this area and trying to help in the medicinal field. You know, if I give you a brief summary, I would say in anti-cancer drug, more than 60% of molecules are from natural product. However, since 1990, the pharmaceutical company have started thinking about curtailing down natural product research. Curtailing down means they were not ready to invest more in natural product research. The reason, there are several reasons in my subsequent slides. I think we will discuss them in detail. But but the output of this is that nowadays the pipeline of these pharmaceutical companies are getting dried up, and the new scaffolds are not available, new therapies are not available, only new generation of the same scaffold is available. So that is an alarming situation, and I think that is why the natural product research and their new advancement in natural product research is very important topic of research, as I would want to highlight here. One more thing I wanted to show to all of you, if you are not sure about it, maybe some of you might be knowing this, that drug discovery and development is not a simple task. It takes more than 12 to 15 years from hit identification to a molecule in the market. Huge investment also needed for this. So if you can see my slide, sorry, if I go back, there is 1 second. Yeah. Okay, let me minus this. Yeah. Okay, if you can see this slide, you can clearly see on the right hand side this flow diagram that several compounds from various sources we generally screen. We find out the biological target, we validate that, we optimize our lead. Preclinical data studies that needs to conduct on animal models, and then we have a good data in terms of minimum toxicity on animal models and sufficient efficacy. Then only it goes to the clinical trials, and which is nothing but the first in human trials, phase 1, which looks after the safety part, which on a small number of volunteers conducted on the healthy volunteers, which they give a small amount of drug and see the hazard or see the safety perspective of that drug. In phase 2 trials conducted on the patient and small population and see the efficacy mainly, and also the safety. In phase 3 of the large population, and the more as you are going from phase 1 to phase 3, the amount that you need in terms of money is exponentially increased. Then the FDA approves this particular data received from preclinical to clinical, all the data, and then they approve it for certain population, for certain treatment. And then what we see that, okay, US FDA and other EME has approved so many drugs in this particular year. But that would have started maybe 10 to 20 years back, that process. Now, like here I mentioned, one new drug would cost maybe 1.5 billion US dollar. That was the available data, and based on that I'm mentioning that it's a huge amount of money. And someone like the investor and all, if they want to put that money, they want to make sure that they get a good return. Definitely the blockbuster drug are getting good returns also, and that's how their discovery, their NC research are in progress. However, the new companies are definitely, this is a big challenge, and are definitely will think twice before they invest in this particular area. Now, this is already a complex process, right? As you have already explained. Now, in that, if you talk about natural product-based drug discovery, which is again further complicated, because in drug design molecule, when you design a molecule for a particular target, you know the target already. But in nature is not producing any molecule for this particular target or for our medication. There are different role of these molecules in nature, right? This secondary metabolite, what we talk about, small molecules also people are referring to. So now when you don't know exact role, or when you don't know how this molecule is going to use for our use, then it is difficult to connect these two things. This is a big task. Generally, people follow bioassay guided isolation. Bioassay guided isolation strategy is, if we are looking for anti-cancer molecule, we check in the extract level, in a fraction level itself, that okay, this extract or fraction has that particular activity or not. If it possesses that activity, we go further in terms of isolation and isolate and identify the compound. It is a lengthy process. It takes more than 6 months to 1 year to find a compound which possesses that particular activity. And therefore, there are several bottlenecks or challenges, I would say, for natural product-based drug discovery. Now, the first challenge I am highlighting here is the isolation of known compound. What I mean by known compound is that, suppose in India, in our lab, like Ahmedabad here, let's say, we have isolated a compound from plant XYZ. Now, when we isolate the compound, we understand its structure, and when we check that if anybody has published this structure already or not, and if the answer is yes, it is kind of a futile exercise, unless and until you have a very good, significant improvement in the biological activity. Otherwise, it is difficult to even publish that molecule. And so that is one of the major challenge, and the people, and this is also the challenge faced by most of the scientists all over the world. Now, that's one of the challenges. The second challenge is the large-scale isolation. There are several bioactive natural product molecules. They are in the shelf in the labs of different scientists, natural product chemists, because they could isolate only in milligram level of those compounds. They couldn't get in grams or kilograms level, because when you want to perform preclinical or clinical studies, you need a good amount of this molecule, good quantity. And if you want to market that, you need to have a sustainable production method for these molecules. That is also a big challenge, by the way. And the third, which I have already briefed you about, is the biological target identification. Because these molecules are in the nature, and if you observe, there is most of the discoveries are the serendipitous discoveries. And that's the reason finding a target for a molecule is also a task, I would say, and it also takes a good amount of time and money. Now, there are challenges, and because of these challenges, the industry, pharmaceutical industry, are hesitating to put the money, invest their research in this direction. But the downside is that if no one invests in this particular research, then after a few years, there won't be any new scaffold, because there was study conducted, and the scientists have found out that the imagination power of human being is based on what they have seen earlier, and that is only the small modification they can pursue or they can think based on their available knowledge, not drastic different from that. And so that is, I would say, is a big thing. And so someone needs to come forward and take up these challenges and solve it. And that is the problem statement right now in natural product drug discovery. The ideal would be academic labs and national institutes should play a major role here. And our also approach is the same thing, that we want to contribute in this area through our research. As I'm mentioning here in this slide clearly, that on the top part is how this process is complicated, that from plant material or any other source, natural source, you extract, you fractionate, you find a compound, you get the compound, you isolate them, characterize them, you kind of find out the activity, and then you come to know that, okay, after 6 months or 1 year or so, you isolated the same compound which has anti-cancer potential or anti-diabetic potential. Then you say that, okay, I have wasted almost 1 year without getting a new compound or anything. So, when these things was there in the industry and academic going on, parallelly there was an improvement in the analytical tools and analytical understanding. Hyphenation techniques were there, they evolved. Hyphenated technique means you are combining HPLC with mass spectrometer, that is called as LCMS. You are combining LC with NMR, that is called as LCNMR, you know, and then several others also, GC, GCMS, CEMS, and all other techniques are there where there you are combining two things and then getting the information that independently those techniques couldn't give the information alone. That was something which was very important. Now that process the scientist has used in natural product research, and that is the term that is coined as de-replication. De-replication. We don't want to replicate the work that other person has already done by using different tools. So though the name is de-replication, but there are different approaches within that strategy, you know, are there which can be followed. Basically, if I want to tell you what is it, it is a process of quick identification of known compound, of known chemotypes, which are already published. How we do that? So here, in one of the published paper, they have mentioned it as a bag of tricks. You know, tricks means you are trying to do some kind of quick analysis or quick magic and to say that, okay, this is there inside of this. It is not the magic, by the way. It is just an analysis. It's a quick analysis. And that analysis has been carried out with these analytical tools, which are very important. You know? So if you see this diagram carefully, what we have natural product source. Suppose you have collected plants, 100 plants from different area of India. And now you want to see that which plant extract should have worked first. You need to prioritize it. 100 at a time you cannot work. Now how will you select? How will you decide? And that is where these things play a role. There are bag of tricks which I said here, maybe n number of things, and the mostly used technique I would say is LCMS based, LCHRMS based. LCNMR also has been applied in several labs, but the cost is a little bit higher there. So there is also a trade there between the cost and the application. Now, if you observe, if you analyze, and that says that, okay, no, this compound has been reported by some so and so person from this and this plant, you know, then you don't work on that extract anymore, because you know most of these compounds are already reported by these scientists already. It is published, and they also have reported that, okay, this is the activity of those compounds. Out of 100, let's say you have came across 5 plant extracts where you have not got any hit from the reported database. Then you say that, okay, would you agree with me that your focus would be more towards those 5 things where no one has worked on it, and the chances of getting novel things would be more in that. And so that is the reason, and that can be analyzed. Just you make a fraction quickly, you extract it and make a fraction, or you can analyze the extract as it is. It can be done within 1 to 2 weeks. It depends on the facility what you have. With a very good facility, you can do in a days. And so, rather than spending time like 6 months to 1 year, you can definitely spend 1 or 2 weeks and to find out, should I work on this extract or not? And that question is very important to ask. To save a lot of things, to save money, to save efforts, to save the field, research field also. Because if you produce more new compound, I'm sure you will have more confidence in that research and all these things. There is one statistical data, as you can see on the right hand side of this slide, that the number of publications in different years from different countries. And as you can see that from 2004 to 2013, actually this review was published in 2014, and that was interesting review, but after that I didn't get the data related, but I'm sure the number of publications would have been increased tremendously. As you can see that in 12, 13, number of publications on de-replication was like double than their previous years. And so you can see there was very well acceptance of this particular methodology in the scientific area. And that is what I think I wanted to highlight here to all of you, that how we are doing here in our labs and how that has helped us to, I would say, to improve the process. Okay, this is a simple definition which I have got it from the dictionary of the de-replication, which I have already explained it to you, but it is really clearly mentioned that it is a process of testing the samples of mixture which are active in the screening process to recognize and eliminate them from consideration, right? If we are already studied, if they are already studied by someone else. It is a stage subsequent to the preliminary screening in the process of discovery of new pharmacological active substances in the mixture of natural products. So by applying this process, by applying this strategy, you select the extract or plant specimen for which you want to work further in detail, such a way that you will end up with most of the time a novel compound at the end. All right. So if I move forward, what are these different techniques and what are different methods? These are the few of them I have mentioned here. Earlier it started with simple HPLC with in-house standards, where they used to record the retention time, UV profile, and that they used to use for the identification. Then later on, when the LCMS came into the picture, they have started using mass data, mostly the accurate mass when it came. Then the process has got a really good strength, as the databases, worldwide databases, common databases can be used. And we will see in the next slide. There are a few other methods, like I have said, LCMSMS, LCNMR, LCMSMSNMR, and many more can be used. GCMS is also one of the techniques which can be used and which are being used for identification of this compound, for the volatile compound, right? So what are the different strategies, I would say? And what I mean by strategy, I just wanted to quickly give you a highlight of this. Like I said, it's the early identification of the molecules and the instruments which are like LCHRMS, LCNMR, LCMSMS, and all. Now, if it is MSMS or LCUV, we generally use our own databases, where we create these databases with the compounds which are already isolated. Now, if you know the accurate mass, and I will have an example in our subsequent slide how we use that, I will show you. That data is being used, and you can search that throughout the different databases which are available online. Some of them are available freely, some of them are the comprehensive one. People have spent a lot of efforts to create those databases, so they are charging for the use of that. So you need a database. You have created some data and then you are comparing it with something else. You need to search, right? And so that is what the database are important. Now, in a dictionary, if you want to find out a word and it's a hard copy, then it is difficult for you to go to alphabetically and then find out that word, and then you get the meaning of it. But nowadays, in the Google Google era, you can simply search and then get the meaning of that word very quickly. How it is possible? Because you can search it. You know that search engines are very important. The same thing is here. If I can search that, okay, this molecular weight, this compound with a molecular weight, this and this, and from this and this plant, Is anyone has reported any molecules earlier? And then you get the data, yes, 3 molecules for this molecular weight has been reported from this plant. Will it be used for your research? Definitely yes. And so that is how we want to use these kind of tools, and we want to speed up our research. We want to focus on something which is different, which is new, and which can contribute to this area, this research area, to be honest. Now, let's discuss about the strategy part quickly. So in this case, what we have fractionation of crude extract, then we apply this de-replication analysis, and then we screen for bioactivity. Then we get the active NRE. So for example, here you have 100 plants, you have de-replicated, you selected 5, and then 5 you have conducted activity, and then you then perform the bioactivity here. In another way, imagine that you have kind of changed the second position of de-replication to the third position. Replace this. So now what will happen? All 100 you have to test the bioactivity. Then the bioactive ones you will kind of perform the de-replication, and then you go further. What are pros and cons? What is so fussy about? Why we are so fussy? Because they give different information altogether. That is why the strategies are. So strategy means what we are looking for and how we design our protocol. Suppose I am looking for an anti-cancer molecule from particular, let's say, fungus. Now I have 100 fungus in my lab. I have screened their secondary metabolite. First of all, I need to know if there is anti-cancer potential or not. So what I'll do, I'll prepare extract, I'll check that if these molecules are showing anti-cancer potential or not. And then I will de-replicate to see that what are those compounds? Are they known? Then I'll ask, are they known for anti-cancer? And if the answer is no in any one of these questions, I will go ahead and isolate those compounds. While in the other case, if I apply de-replication, all the extract, whichever are the known one, I will eliminate that immediately. There might be anti-cancer molecules there also. So you are getting a different extracts at the end. So these kind of strategies are very important and should be very thoroughly analyzed and put forward thoughtfully, because the end product will be different if you apply different strategies. This is just one example I have shared with you. There are n number of possibilities and you can do with this kind of strategy. This is actually the point I wanted to make, because the diagram was very small. As you can see here, the very well acceptance of this particular process in the natural product research, and which you can see the bar diagram very clearly that the people have reported various strategies and various I would say de-replication processes tools during these years. These are some of the publications in this area. So it is not only related to the drug discovery. It is also, you can extrapolate to some other areas also. So for example, this is related to metabolites. This is again related to new compound, microbial research. Well, this is food application. They are using this strategy to find out the compound or metabolite which is present in this particular maple extract. This is also one of the applications. The tool is same. You are trying to find out the molecules. So here they have used this and they wanted to see how many lignans are there, flavonoids are there, maybe other tannins are there, and they have analyzed that accordingly. Data, which was also an important task in food application. Now, with this background, I have spent a good amount of time in background, because I wanted to give you the importance of this particular tool and technique. And at the end of this today's discussion, today's lecture or talk, I want you to at least take this message at home that okay, there are new techniques are available and that can be used to find at the early stage the nature of molecule, identify the molecule tentatively with a good amount of confidence limit uh, through analytical tools, and need not to isolate if they are known. You can have a different strategy to get that. So, like I said, that the three different bottlenecks we discussed earlier, at least if you can solve the first bottleneck up to a certain extent, that would be a good contribution in this area. And that is what we are doing. I have, in my introduction, Dr. Mugan, that I have worked several years, like my PhD is from marine natural product research. So I worked on this particular coral when I was doing my PhD. And I have also worked on various plants in the Piramal and other organization, and also microbes. Now, microbes are very important, and nowadays I have moved, I have shifted. I'm still working on some of the plant, but I've shifted gears and I'm more focused on the microbes now. And I'll tell you why. Because there was a second bottleneck, that supply issue. The sustainable production. If I find out a novel compound in the coral, for example, and which we did, uh, but it was difficult to get that compound, for the large scale production. Same applies to the plant. This I put it for the case of the Taxol also, it was very difficult. That's why it took so many years to develop that molecule. Because the large scale production of that particular compound is very important. Now in the microbes, in the case of microbes, if you have that microbe in your lab, it might be fungi, it might be bacteria, actinomycetes, algae, you can produce them in the lab by fermentation process. You may have a different type of fermenters, different volume of fermenter. And then you isolate those and you can have, literally you can produce a mass quantity. And I worked on one project which went to up to the IND level, where we performed the studies on the clinic, just before the clinics, first in human trials, we have conducted all the studies. And that molecule we have isolated up to 1 kg from natural product. So that's why I'm saying you that from the microbes we can do that, and that is why these tools are very important. Now these are some of the machines that I have used during my research, and they are really automated, and that's why you can speed up the process. We could do this job in 2-3 days because of these tools, which here I am showing you one of the liquid handler, which can in fact inject the sample into the column and then can collect it, the fractions, and you can simply, the track we can put it in the evaporator, which can dry it for you, and then you test the bioassay 96 well plate through high throughput screening for different type of acids such as anti-inflammatory, anti-cancer, anti-infective. Used to do it separately, microbial assay, but at least some target-based assays and some phylogenetic assay, that phenotypic assays. Phenotypic assays can be done through this kind of technology, and where we can speed up the process very nicely. This is also one of the setup that we used to have. That looks a little bit shabby, I would say, the HPLC and the fraction collector, but that is how the labs are. If you are running 24/7 and they are at several connections, and then you are trying to collect all the information in one analysis. And so that is how we use. So we had an HPLC, we used to use a good amount of sample in that, and 90% used to go to collection. Fraction collector was connected to it through here. And that fraction collector we used to submit for the bioassays. And so we knew that, okay, at the time of, let's say in this case, if I'm not wrong, around 18-20 minutes, we have got a good bioactivity, and at that time, what was the compound present, what is the mass analysis, what the database talk about mass analysis. So for example, I'm seeing the anti-cancer activity against some particular cancer cell line. I found this active, and I found that, okay, this compound is already known for the anti-cancer assay. Period. I would not work on that extract because I know that this particular extract has this compound which is showing this particular activity. Now we are all the dots connected, because the extract has activity and most probably it might be due to this compound which is already reported for this particular activity. Now the same compound is present in the 10 extract, for example. Then I have that cluster analysis also grouping, and that can be done easily with one of the instrument which is referred as UPLC, which is little bit advanced than HPLC, where we can have a good speed, and that is like ultra high pressure liquid chromatography that is people are referring to, and that can be connected with the smaller columns with the lower particle size. And so the efficient separation, the same kind of separation or better separation in a small amount of time. So earlier maybe 30-minute runtime, here can reduce to 6-minute or 7-minute runtime in this case, right? And so we can cluster them, similar profile, and then we can group them, or we can call it as a chemical fingerprint which is present in that extract, and then we can club, and then we select one of them based on the yield. We had different detectors here like LSD, UV, and mass detector connected to the one UPLC. And so all the information about that particular sample we used to get immediately, very quickly in one analysis. So that is how the things needs to be carried out if you want to speed up the thing and you want to focus on the project which will end up with the most of the time with the NCE, or what we call as new chemical entity in drug discovery research. This is one of the projects referred as a PP08100173 that we worked in the Piramal, and there we have selected this as the large scale. After de-replication, we found some interesting masses and interesting compounds in this fraction, and so we went ahead for the isolation, and then we also performed some kind of enrichment strategy here, where we have kind of enriched the compound which we were interested in, that was considered as a novel based on the de-replication strategy. And so we enriched them in particular 40% enriched fractions, where we could get the maximum compound in this. And then we further developed analytical and preparative HPLC method, where I would just want to highlight here the reason, because some of you may not have exposed to the preparative HPLC. Generally in HPLC, if you have used, we generally use the flow rate of 1 ml per minute. While in the preparative HPLC, we use 100 ml per minute flow rate. The column generally is 50 mm column, while our analytical column is mostly 4.6 mm. So 4.6 mm column is like this, small column with this width, while the preparative columns is like a dumbbell-like shape. You have a very good amount of width there, and so that you can load a good amount of compound in that column, and you can collect a good amount of this. Because when you inject 100 ppm, that is 100 microgram per ml, and if you are injecting, let's say, maximum 100 microliter, so you are injecting only 10 microgram in injection, and that 10 microgram, if you collect the compound, you will get in that microgram. So you need to inject more quantity so that you can get a good amount for the after purification. And so same thing here, we used to inject 1 gram of compound after enrichment, and that is how we used to collect a good amount in 1 or 2 run injection itself to get the good amount of sample or compound in a pure form. So after purification also there are several steps, and I wanted to quickly highlight those. That we generally go with the purity check. When we check the purity, we check the profile also, UV profile, and then we see the fragmentation pattern. For in-house library also we collect that, so that once you know the structure in-house, if you get the retention time, you get the mass information, you get the MSMS information. It is easy for the next sample to analyze it very quickly. Then the structure elucidation process, which is also a very big area, I would say. And in analytical tools, structure elucidation, I would say, is as a cream, a pinnacle. And you know, that is the toughest as well as the interesting part of any research in the natural product area. Sorry for the typo here, that one should be superscript and 13C should be superscript here and not this. And however, these are some of the two-dimensional NMR technique and one-dimensional NMR, where we get information about the protons and carbon environment, and we assign the structure based on this data. It's a big area of research, and you know, I generally teach this for our master's student here for a complete one semester. So it's a big thing. It will be difficult to explain in one short this 1 hour talk. But just wanted to give you an idea that the proton and carbon NMR and this NMR technique has given a very good advantage, because you know, because of this we don't destroy the sample. At the same time, we get the structure of this compound, and that is very important. Unless until you don't know the structure, it is difficult to proceed further in this direction. And these are some of the other things. By IR, we get to know the functional group present, MS and molecular weight, molecular formula, you know, purity estimation. HRMS gives the molecular formula. I have an example for you as a case study. Yes, we will have time. I will try to complete in next 10 minutes quickly, so that we can have a time for the structure discussion part. If you have any question. So there were several projects that we conducted in this area and found out several NCEs. When we were at the earlier in the industry, we used to get 6 to 7 NCEs per year, and we have also conducted one DBT sponsored project, mega project, where we have screened thousands of extract from different part of microbial extract from different part of India, and we arrived with selected several microbial extract that are we deposited at NCCS Pune, and now we actually got prepared a catalog of compound, and we have isolated some of the very potent molecules and selected NCEs, and that we have handed over to DBT during this project to take it further in the development for those molecules. So to give just a brief about it, that what were the different strategies we used. So like when I was doing my PhD, we used LC-MS-MS based strategy, where I used to create a library in-house by using the ion trap instrument, and where we have created a fragmentation of the particular molecule. And based on this strategy, we have de-replicated several compounds, and we have isolated also several new compounds, which we published in Journal of Natural Product, which was one of the well-renowned journal in this area. Okay, so this was another case that I wanted to show, which is a recent one that we are working on, one of the Indo-Sri Lanka collaborative project, which was funded by DST. And there our hypothesis was to find a novel compound from lichens present on the mangrove plant. And these are the mangrove plant. This is one example, which is Avicennia marina, which is from Gulf of Khambat region. And this is actually lichen, which is a different lichen, but we have also collected another thing from this. So this lichen is like a symbiotic organism between the fungi and algae, cyanobacteria. Now they are symbiotic organism, and you know, it's the complex thing. So it is not a symbiosis between these two microorganisms, but there are another microbes, they reside inside this lichen heli. And we wanted to explore endolichenic fungi. Means the fungi which is not forming this lichen. Because you know, to grow this lichen in the lab is again a difficult task. We don't wanted to work on something which is a large scale or sustainable production would be difficult. So we wanted to work on something which you can grow in the lab in the large scale. So we wanted to work on the endolichenic fungi which reside inside the lichen and can produce this compound for us. As you, in the marine ecosystem or this kind of mangrove ecosystems where there is a lot of competition for space, survival is difficult over there. And so these microbes produce a lot of chemicals. So chemical warfare is there. And that's why they are a good source of novel chemical entity. And that is the reason that we have selected and chosen for our research. Sri Lanka is the best place to get the lichen, because of that island and that environment. There were so many lichens. I visited Sri Lanka University of Kelania and I could find in the campus itself, there were so many lichens in the plant, on the trees, in the small plants also. And so it was the varieties of lichens that we collected and we published several papers from them also there with that. Now with this, we got the endolichenic fungi, we fermented in the lab and prepared the extract. And here the idea to look for the anti-cancer molecule, because most of these molecules are used in the antimicrobial assays and they showed good antimicrobial activity. And generally the antimicrobial activities that turn out to be good anti-cancer activity as well. So in both the direction we can pursue it in the large scale, and we could get a good NCs from this project also. One small case study I wanted to give you in detail for one thing, which is about HRMS, because now you will realize you will understand what I'm talking about. It should not be only superficial talk. You should get some insights also. So, for example, this is just an example from textbook. I have taken this. So there are 4 compounds with the mass 44. If you can calculate, like if you take a 12 as a mass, atomic mass for carbon and 1 for hydrogen, so this total would be 44. Same thing for this. If you add 16 for hydrogen, 16 for oxygen, 4 for hydrogen, 20, and 24 for carbon, because 12 for each, so 24. So that's again 44. Same applies to these also. If you can calculate, you can calculate. Now you see that if you find out with our mass spec that okay, my compound has mass 44, there are so many molecules can be possible. So you cannot de-replicate or you cannot identify like this. However, if you know the exact mass, and that is what is called as that we'll get through high resolution mass spectrometry. That is also referred it as accurate mass. And that can be up to 3 decimal places, 5 decimal places based on your instrument resolution. In this case, let's take an example where we have got 44.029. Now we know the accurate mass of these individual atoms, elements. Now carbon has 12.00, hydrogen has 1.007824, and same for oxygen, nitrogen, sulfur and all other things. If you add those accurately, you will get the accurate mass up to 3 to 5 decimal places. Now you compare the accurate mass with the observed mass, and that is what it is has been done here for these 4 molecular formula. And it was observed that this was the closest one with this observed one. And so now you can pick the closest one, the closest formula for our compound. That could be the compound of which is present in that particular action. And that is how we kind of narrow it down the probabilities, and we get a good success rate at the end for identifying this compound. I just wanted to give the power of this high resolution instrument that you can simply identify the molecular formula by doing this. And for this, you don't need to isolate that compound because you are separating it in a chromatographic HPLC column, and then you are sending one by one to mass, and that is exactly That's the beauty of this work, and you can get the best of both worlds, where separation is there and where you get the high resolution also in that case. So this is another example also, the precise mass you can get, and you can assign the molecular formula, right? And by doing that, I would say this is really advancement of the analytical techniques have helped us to proceed further in this direction. We are working on several plants here, and we have worked on some of the projects related plants also. So that is also important, because there are several studies have been carried out from plant specimen, and we don't want the small labs in and around to work on the same thing again and again, and and isolate the same compound. So this is also one of the things that as a national institute we should work, and we should collaborate, and we should take the projects and quickly help each other to find the compound. So with these 4 plants we have conducted the case studies, and we have identified the already known compounds which is already present in that. While in one of the plant we observed, which is a Kachanar plant widely available here in Ahmedabad region, that we have collected also from this. Baunia genus, Baunia variegata, Baunia purpurea are the mostly commonly found plant species. So I wanted to give you case study. I know the time is limited already, 11:44 is there. So I'll quickly, 5 minutes, not more than that. We just wanted to give the both the things, how we work, like based on the bioactivity guided isolation. At the same time, we wanted to apply the de-replication strategy also. So this is generally, we generate HPLC fingerprint, we conduct the de-replication strategy, we identify the compound quickly in the HPLC analysis itself to see that, okay, this can be the probable structure or not. And if you don't find the hit for most of the molecules, so for example, these were the hits that we observed with the database. We have procured dictionary of natural products also, which is not the hard copy but the soft copy, where we can do the search, search based on the molecular weight, genus and species, based on the UV profile, based on the accurate mass, based on the activity, all sorts of criteria that you can feed, and you can get the hit from this. We perform the fractionations like we generally do with the natural product isolation. We enrich the fractions which we wanted to focus on. We wanted to know that, because we found some group of the compound which can be novel, which has not been reported from this plant earlier. And then we perform this particular analysis, and we enrich them before the purification by preparative HPLC at Naipur, Ahmedabad. And then there is something, I think so many people are in waiting room, and I think that message is not allowing me to go to the next slide. Just give me 1 minute. Right. So we have finally we worked on that particular extract, and we performed the preparative, semi-preparative we referred to, because it was not that high scale, the amount was less, and so we used a small column which was 17 mm column, and then we purified the compound. We checked the activity and the structure elucidation also, where we got that molecular formula, and then we are working on NC. We kind of know the scaffold, the structures, but we just wanted to, since we have isolated that amount was very less, and we could get only proton and carbon NMR. But we are working on the large scale isolation of that compound now, and it's almost there. With this batch master student, I think we should get the good amount, and we should get the structure out from this thing. So with this, I will stop here. There are several reports, studies that I can share, and we can discuss further also. But I think I just wanted to give you an idea, the overview of our research, what we are doing, and how these analytical tools have helped us to speed up the process in our research. In the acknowledgement slide, I definitely want to acknowledge my students, PhD as well as master students, Without them, I couldn't have done so many of these projects. And today I have explained you Ashwini's project related to that particular plant. And there are several PhD students which are there working with me on the different projects. So I would like to thank my students and definitely my institute, Naibar Ahmedabad, for the funding and support. Department of Science and Technology for the particular grant of lichen, endolycanic fungi. This is my group. Some of the students actually have left recently. They completed their masters, while PhDs are still with me, and some of the new students also joined, but not the recent one, but yes, the near recent photographs of my group. And thank you so much, all our audience, for listening me, for your patience, and I would be happy if you have any question. Thank you.
Thank you, Dr. Abhijit. The session is open for discussion. If you have any doubts, clarifications, you can ask Dr. Abhijit.
Yes, definitely. Thank you. Anyone?
Any questions?
Hope you understood the concept, and I hope I cleared some of the thoughts, and I hope you should take this take-home message with you about this analytical tools. De-replication. Please remember this word.
Yes.
Okay.
Okay. Thank you, Doctor. Thank you so much.
Thank you so much.
Thank you, sir.
Thank you.
Good morning, Professor Sendila. Professor?
Yeah. Good morning.
Good morning.
Good morning to you all.
Students, we'll continue with our next session. We have with us Professor Senthilnathan Senkotayan. He is an associate professor and principal investigator from Sri Paramakalyani Center for Excellence in Environmental Science, Manonmaniyam Sundarnar University, Tamil Nadu. He did his PhD in animal science, biopesticides and insect toxicology in 2000 from Bharatiyar University, Coimbatore, Tamil Nadu. His research interests are insect biodiversity, insect-plant interactions, biopesticides, and environmental toxicology. He also served as an assistant professor in the postgraduate and research department of biotechnology in Vivekananda College. And he did his postdoctoral research at the Department of Environmental Engineering, Chonbuk National University, Republic of Korea. And he worked as a research scientist in Plant Environment Division, Honnam Agriculture Research Institute, Republic of Korea. And then he joined, he was also serving as a visiting professor at Louisiana State University Agriculture Center, USA. And he joined the Manonmaniyam Sundarnar University on 2007 and working there till date. So some of the important awards to his credit are BIIS Fellowship from DST, Government of India, and then Young Scientist Award under Fast Track DST, Government of India in 2010, and Young Scientist Award under RGYI DBT, Government of India 2009. He also serves as an associate editor and subject editor of Nature Scientific Reports, Archives of Phytopathology and Plant Protection, Journal of Asia Pacific Entomology, Florida Entomologist, Biopesticide International, International Journal of Pest Management, Frontiers in Invertebrate Physiology. He has secured research grants of 223.59 lakhs so far through various projects, has guided 2 postdocs, 12 doctoral theses, 3 MPhil and 37 master students. He has published 3 book chapters and has more than 67 research publications in international journals. He also serves as a funding agency advisor to Czech Science Foundation Republic, Czech Republic, Department of Biotechnology, Government of India, Department of Science and Technology, Government of India, Council of Scientific and Industrial Research, Government of India, ICAR, Government of India, and Ministry of Environment and Forest, Government of India. With this short introduction, I welcome Professor Senthilnathan to deliver his lecture on plant-based biocontrol agents for sustainable agriculture. Welcome, Professor. Your session starts. Okay.
Yeah, it's a very good morning. Yeah, right. Good afternoon to everybody. So it's a very great pleasure to be here, and also because they're providing the resource to the students and the budding scientists is always be enthusiastic for us. So especially for nature-oriented work and research to be implemented with that ecological aspect is always be very interesting, and also it is very essentially needed for current scenario. Hence, the topic is very important for eco-friendly aspect to protect our environment. So with that, I'll start my lecture. Can I share my PPT?
Yes, Professor, you can share.
Share the screen. Yeah, okay. So right now it is sharing, right?
Yes, Professor. It's visible.
Thank you very much. Okay, so my topic is plant-based biocontrol agents for sustainable agriculture. So before entering into the topic, we should know about that why this is needed for current scenario. Because you know that India is basically an agriculture country. Majority we are depend on the agriculture product and vice versa. So because nowadays the low agriculture productivity is because the land is shrinking and the urbanization is increased. So as a result, poverty, food insecurity, and malnutrition happen there, and population explosion is very huge nowadays, nearly 1.2 billion. Instead of absolutely poverty in this current scenario, and decreased availability of cropland, as I said before, major implication of food security, and accelerated public investment is important priority. Facilitate to the agriculture growth is paramount important, right? So sustainable agriculture is prime most important for, so within the very limited land, we should need a sustainable agriculture. That is probably, here I would point it out, 40% of the crop can be lost into insect damage, because insect is a huge one, which occupied nearly 80% of the earth in various forms. So, and majority of them are non-beneficial, are destroying the plants. The larval stage, especially of moss, especially in the category of Lepidoptera, which is economically damaged the valuable crops. And nearly, we have using a broad spectrum of chemical pesticide and the primary control for the agent of agriculture pest. So there are so many problems by using these chemical pesticide, because you know that there are lots and lots of companies dumping millions and millions of money for producing the pesticides. So economics here, there are some statistical point you should know. Loss is nearly about 64 billion US dollar, and nearly a round figure of 243.4 billions. Increasing the crop production is minimize the pest association loss, estimated 40% as cross loss. So always we are depending on the pesticides. So I would say that there are a lot of companies, including Monsanto, DuPont, Bayer Agro, and several companies are there producing several class pesticides. So the farmers are always believing the pesticide have a great effect on killing that insects. But they know that. But the thing is, always be decreasing the lifespan of human being. That is inevitable. The point is, there are using of these classical and different type of chemical pesticide leads to a lot of problem, including number one is insect resistant. So the insect get modified its immune system. So to overcome the pesticide problem, and then toxicity leads to the ecological imbalance problem and creating the environmental pollution. So application of pesticide, point source contamination, off-use contamination, an atmospheric problem, water resource contamination, including groundwater problem. So these are the several different type of pollution caused by this chemical pesticide. Apart from that, we are using organophosphates, organochlorides, and neonicotinoids, which recently developed. These are all having a problem to the human beings, not only human being, the non-target organism, and also all the domestic animals, including birds and mammals, right? So organophosphate, which kill insect by interfering the activity of their brains and nervous system. So it is a contact poison, having a problem with that nervous system. And organochlorine as like that, very persistent when compared with all these two pesticides. Always be persistent. The cell half-life of this pesticide is very long. And biomagnification, that means up to 4 or 5 point source can be identified. Thin shells of bird cells accumulating even in mother milk. Human endocrine system is damaged due to the organochlorides. Neonicotinoids recently developed by the several companies, including Bayer, Agro, Syngenta, and DuPont, interfere with the pathway of common in the insect nervous system, also which interfere the human nervous system itself. And recent research, which published in the PLOS One, has clearly pointed out that the honeybee population is tend to decrease the use of neonicotinoid. You know that honeybees are a great potential pollinators, so which always be beneficial for the external fertilization or external pollination. So this honeybee colony is very important. Significantly, mortality has been observed while we are spraying the neonicotinoid in the field. So this is a point which here we should know that. So here, alternative pest controls. Definitely, we need an alternative for the overcome these synthetic insecticides, because we should have to point out the clear-cut problem, overcoming this problem. Hence, we need to seek a great solution for that. So that should be eco-friendly in nature. To adopt the pest management strategies, reduce the development or resistance to the synthetic insecticide, we are seeking, or we should seek alternative pest control strategies, that is including natural enemies like predators and parasites, and pest-resistant variety of seeds. And third point is biopesticides, nothing but a biologically originated pesticide which does not create any scume to the environment. That is very important. Even though it has a limitation, but the benefit is more. Mass production, slow rate of action, cost effective, timely availability, narrow spectrum activity, and biologically degraded within very short time. So that is a plus point we should have to consider in this time. And here, there are components of pest control. Before entering into the plant's resource, we should know the branches of this pest control, including biological control, cultural control, chemical control, IPM. I explained previously about that chemical control, but these biological control and cultural control are eco-friendly, especially using parasites, predators, and baculoviruses, entomopathogenic bacteria, fungi, nematode, like that. So the cultural control, including nutrition management, crop rotation, and depending upon the climatical condition, and field sanitization, along with the tillage, showing, and planting density. So the IPM is nothing but integrated pest management that will combine one or more component in the pest control property, that is biological and cultural and plus little chemical. So here, I'm not seeking for complete eradication of synthetic pesticide, but I insist to decrease the chemical or synthetic insecticide usage. That is the point. So major agriculture pests we are focusing here: aphids, caterpillars, cutworms, and low-caste mites, thrips, spider mites, whiteflies, snails, sliders, weevils, bugs. So these are the important, significant organisms which having a problem with that agriculture crops. Here, the point is botanicals. So here, why botanicals? The botanicals are renewable resources. This is available, rich resources of plant diversity is available in Indian subcontinent. That's why we are focusing on botanicals. The use of plants, plant-based material, or crude plant extract or pure compounds to protect the crops and stored products from the insect pest. As old as crop protection itself from the, because this method was identified nearly 5 to 6 decades before. So in not only 6 decades, in olden days, our ancestor used such methods, especially crude methods. So prior to the development and commercial success of synthetic insecticide, beginning in the 1940s, botanical insecticides were major weapons in the farmers' arsenal against crop pests. So the golden time should come back right now. So botanicals, the positive point is quick action on target pest, broad spectrum of action. That means multiple mode of action, biodegradable, ecologically acceptable. That's the point. Eco-friendly, safe to the beneficial or non-target organism. So plant-based material or botanical insecticides does not create any problem to the environment. As a result, the biological control agents survive. That means including predators, parasites, spiders, and birds, whatever it may be. So floral resource for natural enemies are always be available. Here, synthetic pesticide and botanical pesticide comparison is there. So continuity to be used. So accumulation always, frequent application is needed, and kill and toxicant is persist. Here, human health, environmental safety, and market access, sustainable agriculture, all are feasible in this methodation. That means adaptation. So takes a days to degrade. Eco-friendly, no toxicity to the non-target organism, including the farmer's friend. That means earthworm, ecologically indicator animal. More frequent application is also needed. That is a somewhat negative point. And morphological and physiological interferes appear, growth regulation appear, repellent also available, and kill finally. So hourly pre-harvest interval is needed. Even though that insect survive, the insect is unable to reproduce or metamorphosis. So these are the point, a beneficial point when we are seeking for a botanical insecticide. So here, major botanical pesticide available in the market in from the 1950 onwards. So pyrethrins, rhododendron, and Sabatilla, Rhynia, Nicotine, D-limonene from Neem, and Linalool and Neem. So these are the very limited LD, that means lethal dose, and even with the dermal dose. But these nicotine only a very danger. So here, rhododendron occurs in the roots of Lanchocarpus species in South Africa in olden days. So commercially, rhododendron was at one time produced from the Malaysian dairies. Currently, the main commercial source of rhododendron is Peruvian Lanchocarpus. That is available always, right? So throughout the year. And mode of action is always mitochondrial disinfection. And also necrosis is the second most important, cytoplasmic and membrane damage. So the basic metabolism impaired while you spraying a particular type of botanicals. Second point is pyrethrum or pyrethrins, pyrethrum and pyrethrins, insecticide extracted from the certain species of Chrysanthemum family. It's commonly available in Indian subcontinent. There are two most important chemicals identified from these pyrethrum, from the Chrysanthemum, that is pyrethrins and pyrethrum. Pyrethrins are the 6 active molecules or esters that act as a killing agent in the extract. Pyrethrum is the total extract from the flower, while pyrethrin are refined as a 6 excess, a pure compound. Here, that targeted with that Chrysanthemum pyrethrinoid, that means targeted with mosquito, flea, flies, moths, that means multiple mode of action with multiple insects. So here, pyrethroid modified the sodium potassium pump. So the action potential is very important for the locomotion of any animals, including human being. So any impaired in the sodium potassium pump, which leads to the which leads to the break the locomotion of a particular animal. So as a result, the total metabolism is get affected. So that is a point in that particular pyrethrinite. So here is nicotine, right? You know that everybody knows this nicotine. Actually, the plant Nicotina tabacum was identified the purpose of pesticidal activities in olden days because it is majorly cultivated in the North American scenario. And later on, human beings are identified which having the nicotine content, which giving so many enthusiastic effect in the human body. So they are modifying with that dust for the cigarette and modifying for chewing purpose. So these are all very dangerous to the human health as a result, inducing the cell cleavage. That means increasing the cell division as a result, cancer at the result because it contains the pesticidal activity. So that nicotine is a simple alkaloid derived from the tobacco that's called the Nicotina tabacum and the Nicotina species. Nicotine constitutes about 2 to 8% of dried tobacco leaves. Insecticidal formulation generally contain nicotine in the form of 40% nicotine sulfate, currently imported in small quantities from the India for the pesticidal purpose. So mode of action of pyrethrin combined with the nicotine disturb the sodium and potassium ion exchange process in the insect nerve fiber. So as like other organophosphate and organochloride. So it interrupting the normal transmission of nerve impulse. So the nerve impulse is very important for the locomotion purpose and extremely fast acting and cause an immediate knockdown. That means immediate paralysis to the insects. So here the diagrammatic representation of DDT and also other agents. So mostly these acetylcholinesterase inhibitors. The second most important tree or rather plant which having that means from the root to the inflorescence, from top to bottom, all the plant parts of this particular plant has insecticidal activity. Not only insecticidal activity, all other biological active compounds fulfilled in this plant. So neem produce, that is technically known as Azaracta indica, are derived from the neem tree Azaracta indica that grows in the tropical and subtropical region, originated from the Indian subcontinent. Even though several countries are having a patent for that particular plant, so we overcome the problem, fight against that patent. Now we revoked all the patent obtained in the neem plant. So we are free to do great research on that neem plant. So the principal active compounds present in the neem is azaractin, a tetranatriterpene, chemically known as a bitter complex chemical, both feeding deterrent and growth regulator. Silanine and other minor components in neem also having a insecticidal activity. Neem product including teas and dust made the leaves and bark extract from the whole fruit seeds and seed kernel, and an oil expressed in the form of seed kernel. So that is primarily this azaractin can be extracted from the neem seed kernel, dried neem seed kernel. Here, so multiple mode of action of this Azaracta indica, right? Repellent, pest control, insecticide control, growth regulator, reproductive inhibitor, and also some part of it can be used as a fertilizer. And there are several health point of view, especially in veterinary medicine and also cosmetic and also medicine, that are also controlling the malaria, that means Plasmodium vivax, and also ulcer, mouth ulcer, and vice versa. So these are multiple, more than 100 products developed from the neem, which particularly emphasizes the pesticidal compound, especially companies like Goan, Fortune Biotech, and EIT Pari India Limited, Agrologistics, and Wooden Stream, and EPP Limited of Brazil, and Ozone Biotech from India, and Indian product especially originated from India. So these are the companies which having a different product, which including Azadirect, Neemix, Azamax, and EcoSense, azad-acting neem oil and bio-neem and neem and neem tra. So these are the specific commercial nomenclature of these neem product, especially developed from the neem. And here there are a typical symptom of neem spray against the insect. So molting inhibition, metamorphosis inhibition, larval pupil intermediate, and what can I say, juvenile hormone impaired, and several morphological defect can be identified while spraying the insect in neem-based insecticide. So this is a chemical structure of azaractin. Azaractin. So here some mode of action of neem, that including reproductive effects, neuroendocrine activity, anti-feeding activity, and cellular and molecular effect, that including inhibiting prothoracic hormone and juvenile hormone that known as JH and PTDH, and blocking calcium channel. I said before that blocking the sodium potassium pump as an immediate paralysis or knockdown methodology, and interfering with the IAS pathway. And what are the reproductive effects? We shown that reducing a fecundity, even the viability of egg laid after that is decreasing, and number of egg laid by the particular animal after treating with neem is tend to decrease. A miniature type of insects developed from the egg, and oviposition deterrency happens there, and anti-fedency, including the insect sensitized getting affected, inhibiting the fecundity and inhibiting the feeding. Apart from that, feeding, food conversion, digestion enzymes are impaired. So what is the cellular and molecular effects happen there? Cellular apoptosis and disturbing protein synthesis, and upregulation and downregulation of metabolic protein genes are differ in such a way. So here the overall effect of azaractin against the insect, we know that. So that primary antifeedance, secondary antifeedance, insect growth known as IGR, sterility, and cellular process. So these are the well-known compound identified from the neem, that's called nimboli, jejunine, azaractin, selanin. Apart from that, deacetyl jejunine, nimbin, and so many compounds are available in the neem extract. So target agriculture pest as far as neem-based insecticide, beetle larvae, moth caterpillar, and stalk borer, and brown plant hopper, which is a serious pest of rice, it's called as Neelparvata lugens. And here, second important neem mode of action, I said, partially I divided for the agriculture application, and apart from that, there is some beneficial effect, medical application, including anti-cancer, antimicrobial, anti-inflammatory. So the action against the insect is very nice when compared with the human being. Even the limited amount of neem-based extract can be consumed by the human being without any side effect. So these antifeedant, growth molting inhibitor, and reproductive effect of neem-based insecticide is a unique process. So the insect does not having a power to overcome this problem. So here I said that anti-cancer, immunomodulatory effect, wound healing effect, these are the beneficial effect of that neem. That hepatoprotective and anti-malarial, antioxidant, antifungal, antibacterial, antiviral, anti-inflammatory, and neuroprotective activity apart from this. So this is famously known as the Karpagavrusham. And apart from that, this is a schematic representation of how these neem, especially neem-based compound, act on that insect, especially for physical method, appearance of spray, that means cuticle spray, and sodium potassium channel, which unable to rotate the action and resting potential and alkylating agent, because the gut line is getting affected by these neem-based pesticide. So second most important is Melia azaracta. We know that lilliput, that means there was China berry or Persian lily, which is also invariably available in throughout the Indian subcontinent, that is from the Meliaceae plant family. There are multiple compounds in DIAS like Neem, Azadirachta indica, it is significantly, when compared with the Neem-based Neem, these Melia Azadirachta does have a significant compounds itself, that is having insecticidal, antifeedant, growth regulator, insect deforming, and growth modifying properties. So that including catechin and chemoferol and beta cytosterol. These are the major compound. Apart from that other compound, these are the major compound available in the Melia Azadirachta. So the target pest including sting bug, sucking pest, and rice leaf roller, that's called Neplocrocus medianis, otherwise known as rice leaf holder. And tritoma, that's called sting bug, which is sucking the sap from the plant material. So these two are very serious pests as far as farmers concerned. Third important point is Anthrographis paniculata. These are there are a lot of research has been conducted because it containing the bioactive compound Anthrographilidae. So all part of this plant is having a great potential for that means bioactive compound. 55 N-labatin diterpenoids are available, 30 flavonoids are available, and tunic acid and xanthosine and rare neuratoids are available in that Anthrographis paniculata. So this is also an external morphology of that particular plant in the flowers or inflorescence of that particular plant. So there are compounds which is primarily available in that Anthrographilidae and apigenin and tetramethyloxyflavone. These three compounds are invariably available in that particular extract. So these are the target pests of that particular compound. Calosobrugus, Spodoptera litura, Anopheles. These are the mosquito species. Two important mosquito which transmit yellow fever, dengue, and malaria. So that's Anopheles and Aedes. Aedes aegypti and Anopheles stephens. Then this is a mode of action. So where the gut epithelial cells are getting affected. So these are the effect of Anthrographilidae. So this mode of action, Anthrographilidae always disturbing the gut membrane. As a result, the acidic condition of that particular gut is malformed. As a result, reverse osmosis happen. The insect getting leakage of water content from its body. That's called septicemia. And the second most important thing is mode of action of repellent. So once if you are getting that particular extract, if you are applying into your skin, so because this plant extract is not producing any irritation or any side effect to the human body, especially skin. So it looks like a lotion. So, but it is having a severe repellent activity against this mosquito. So the mosquito sensile is unable to perform its task. As a result, it cannot, it doesn't have any effect on that particular smelling. So that means especially the sensilla, throughout the sensilla, where it insert the proboscis to the skin. So as a result, the insects are unable to locate a particular point because the sensilla is not get ready with that. Once it is getting affected with that particular smell of, that means aromatic smell of particular plant material. So with that points which I spoke in the beginning itself, we have a few conclusions that should be asserted. Anti-insect compounds are accumulated in several species of Myrtaceae, Lauraceae, Rutaceae, Lamellaceae, Astraceae, Faceae, Fogaceae, Gingerpaceae, and Fibraceae. That is called ginger officinalis, which include ginger officinalis, that is a common ginger, and Pipraceae, that is include Piper niger, that is Piper. So these are the important plant family which have a lot of resource itself. And as I said before, which is a renewable resource and does not release any scume to the environment, biodegradable, ecologically acceptable, and also it doesn't have any impact to the non-target organism, including Odonata and mayflies, and which are all ecological indicator. And also it never produced any residue to the soil. So the soil organism, like a bacteria, a beneficial bacteria, and denitrifying or nitrifying bacteria, and other decomposing bacteria and fertilizer, BAM, and especially I forgot that earthworm. Earthworm, which is friendly to the farmers and also which enrich the soil, which are all survive once if you are transferred your pesticide into a biologically originated pesticide. And apart from that, there are countless bioactive compounds has been identified and isolated so far, and also a lot of research has been conducted and contribute to the future development of novel plant-based bioinsecticide for organic food production. So the half-life, that means the lifespan of human beings also tend to increase while you are switching over to the biologically originated pesticide. So the appropriate selection of biomolecules to prepare bioinsecticide with multiple mode of action against the target pest is unquestionably safer, an alternative for the organic food production. So even there are a lot of biotechnological tools has been adopted for the integrated pest management, that including BT, the Bacillus thuringiensis, with the Bacillus thuringiensis gene that is called the cry gene has been identified, that inserted in the plant like rice and maize, and that is also combined with that botanical insecticide, the effect will be severe, and also which is very compatible with that biological control agent and marker associated selection aspect. So these biologically originated pesticides, so-called biopesticide, act like an umbrella to protect the residues from the chemical pesticide. So this is a point here we should consider. So that is called the crop production. So as a result, the student community or budding scientists should come forward to work with that biologically originated pesticide to protect the world. So that is a point here, that the theme of that my lecture is to improve the strategy of biologically originated insecticide, or creating the awareness to the public through the young budding scientists like a student to protect the world, and also protect the security of the food, and also promoting the organic food production. So, and also overcome the insect resistance, and maybe it is compatible with RNAi technology, which is recently adopted, and cryobiological, and also enhancing the more important components of the stored product. So with this point, I'm concluding my lecture, and if you have any questions, please post to me. I'm ready to answer all the questions. Thank you very much for the opportunity.
Thank you, Professor, for the wonderful lecture. The session is open for discussion. Any clarifications, you can ask them. Any questions, students?
So, so the question is not readily posed to me, because not in this aspect of biologically originated pesticide, or not in that particular lecture. You can ask any questions related with that sustainable agriculture for the ecologically acceptable one.
Good afternoon, sir. I have a question.
Please, please.
So now, what is the status of sustainable agriculture right now in India? How many farms have started adopting that form of agriculture?
So actually, yeah, the very good question you posed to me. So actually, the awareness of using a biologically originated pesticide is very low. As far as our statistical point of view, the farmers are throughout the Indian subcontinent, nearly 4% of that pesticide market. That means nearly 4% of the pesticide market has been adopted with biologically originated insecticide, pesticide, fungicide, whatever the sites available. So when compared with the synthetic insecticide, that means organochloride, organopesticide, or nicotinide pesticide, our biologically originated pesticide is very limited, even though the government are producing a lot of subsidy to the farmers, and also research has been emphasized, but slowly the attentions are diverted to the biologically originated pesticide. So that's why we are promoting the bioinsecticide for the usages.
Okay, so thank you.
Any other questions?
You can also use the chat box.
Good afternoon, sir.
Very good afternoon, please.
So I had a question related to sustainable agriculture.
Definitely.
Are there any affordable solutions where or are people using grey water, which is a wastewater obtained from kitchen, for agricultural purposes?
Yeah, okay. So actually, so usually we are encouraging the biological fertilizer. For example, for kitchen wastages like vegetables and other materials. I'm not talking about that usage of the chemical bleaching water. So we are talking about only the organic material, or rather the vegetables and vegetable waters or something like that. So we usually encouraging peoples to make a vermicompost in the kitchen garden. So that means you can dump all the vegetable waters or vegetable wastes and anything else, which is I'm not talking about that cooked material. So the raw material, raw vegetable peel and everything can be dumped and we can use for the vermicompost purpose. So we can always encouraging, which is a biological fertilizer. So the biofertilizer and bioinsecticides are the two important components for eradicating these synthetic insecticides and synthetic fertilizer. So after several weeks, a very good manure can be developed in that particular area. So you can use that fertilizer for the farmhouse or farm plants.
Sir, also sometimes there is foul smell, and so how can we overcome with some household materials?
No. That means if you are introducing the that means earthworm, a different species are available that immediately decompose these vegetable waste, vegetables and peels, everything. So they can, depending upon the number of earthworm introduced in that particular area, you can get a solution from nearby your environmental department or local agriculture department. Now the government are introducing several schemes for the farm, that means kitchen farm. So you can get all the information from that. So you can also have the worm in that particular area, what dimension you have in your kitchen garden, and you can introduce it. And that vegetable peels and vegetable waste can be consumed by the insect within 24 hours. You do not have any foul smell or bad smell onwards. So you have everything, everything is possible to overcome such a problem.
Okay, thank you, sir. Understood.
Okay.
Professor, we have a question in the chat box.
Please.
Sir, can you please suggest me a biopesticides which will kill insect and pests in rapid rate?
Okay. So yeah. Okay, right. So because there are depending upon the economical point, we can get a biological originated pesticide, especially botanical insecticide. So the Neem particular compound azadirachtin is very expensive. You know that. So we are go for neem oil. So I suggest for that can be act on that all type of pesticide. Neem oil is commercially available. So that neem oil, nearly 1 liter is cost about some 50 rupees, right? 50 rupees in local market, right? 1 liter or 60 rupees. So for 1 liter, you can pour into 5 ml or rather 10 ml. So you can use a soap solution, little soap solution or any ingredient which can mix with that. So use it frequently. For example, you can spray this solution of any sort of sucking pest or the pest which is that means chewing pest or sucking pest, whatever the pest. So you can apply repeatedly in the morning, evening, at least 3 times in a week. Definitely you have a solution for that. So the neem based insecticide is the topmost priority.
Any other questions? One more question has come. Sir, can we use raw leaves of neem or other biopesticides having plants to the agricultural land?
Yeah, please, can you read one more time?
Can we use raw leaves of neem or other biopesticide activity plants to agricultural land?
Yeah, definitely. So there are several plants can be used as a manure as well as the pesticidal activity. So for example, Calotropis gigantea, that is a great potential having a biofertilizer as well as the pesticidal activity. But you very well use the dried leaf of neem, and that is a very great for the what can I say, nematodes, right? So these neem based leaf can kill the root knot nematode, not entomopathogenic nematode. That is the point, right? So it is always be beneficial. You can very well use that dried leaf of neem. Apart from that, you can get a lot of plant resources to use as a dry material for the fertilizer purpose, like a Calotropis gigantea, which is commonly available anywhere else in the world, especially Indian subcontinent. So Calotropis gigantea is always available. And apart from that, Datura metal. These are the commonly available plant throughout the area. You can very well use it. It is very inexpensive.
So Datura has a toxic substance in it, and how can we use as a biopesticide?
So see that the toxicity is in different sector. For example, Datura fruit or Datura fruit is having a high toxicity when compared with the leaf. You can use the leaf. The leaf is having a very limited toxicity, and also you can get it a lethal concentration. You can identify the lethal concentration, very minute lethal concentration. You can use a very limited amount of quantity for that. So you don't, we never use that means fruit or inflorescence. We always recommend to use a leaf.
Yes, sir. Thank you.
Okay.
Any other questions?
Sir, what is the optimal concentration of neem oil per liter of water?
Optimal concentration, for example, depending upon if you are using a sucking pest like a brown plant hopper or aphid or like a sucking pest, you should have to use at least 7.5 ml per liter, 7.5 ml to 10 milligram per liter. If you are using a chewing insect like a Spodoptera or common vegetable, you can use a 5 ml per liter.
Okay, sir. Thank you, sir.
That means you have to use a frequent mode of spray. So farmers are always be insist to use a chemical pesticide. They never practice with that multiple spray. That is very important. You can use the neem spray multiple times. For example, thrice in the week, so you can get a better result.
Okay.
Okay, thank you.
Any other questions? If no more questions, we will wind up this session. We thank Professor Senthilnathan for joining us today and elaborating the students of based on the sustainable agriculture and how plant-based biopesticides will have a major impact in the upcoming years in the Indian agricultural system and also throughout the world. We thank Professor once again for joining us today. Thank you, Professor Senthilnathan.
Thank you very much. And really, I appreciated the students for the patience and questions and interaction, everything. And thank you once again. Thank you for your opportunity. Have a great day. And my congratulations to you all and the organizer, and my best wishes to the fellow colleagues and students. Thank you very much. Have a nice day.
Thank you, Professor.
Thank you. Shall I leave, sir?
Yes, Professor.
Thank you.
Thank you. Now, students, that's all for today's session we are having. I hope you all have read my message in our group, WhatsApp group. I've shared some materials also for your reading purpose and also for the discussion in the upcoming sessions tomorrow and day after tomorrow with Professor Anil Gupta. So please be prepared. Okay. If you have any other questions regarding, you can ask me in the group. Thank you.
Hello, sir.
Yes, Amulya.
Yes, sir.
Sir, this week's assignment, sir.
Yeah. Yes.
Sir, this assignment topic is not given, sir.
This week assignment for this week.
Yes. Sir.
Sorry, your voice is breaking.
Sir, for